This video will show you how to approach a slightly more complicated bacterial dilution problem that you may encounter in your experiments. Consider the following scenario. A group of students wants to look at whether a difference exists between the bacterial load found on organic baked salads compared to the bacterial load on conventionally grown ones. To conduct the experiment, representative samples were taken from each of 3 bags of salad greens. 25 grams of sample was added to 475 milliliters of 0.1% peptone and homogenized in a stomacher bag for 3 minutes to dislodge any bacteria attached to the lettuce leaves. 10 milliliters of the solution was then taken and added to 90 milliliters of 0.1% peptone solution. From this solution, a 100 times dilution was made before spread plating 0.1 milliliters of the final dilution onto tryptic soy agar in duplicates. These are the plate count results after incubation. The first question asks about the number of biological and technical replicates. Biological replicates involves testing different individual samples and tests for variability between samples. In our case, this is represented by the 3 different individual salad samples. Technical replicates, on the other hand, involves testing the same sample multiple times. It tests for accuracy and variability of the experimental method. In our case, this is represented by the duplicate agar plates. To determine the total dilution factor, let's first take a look at the individual dilution steps to calculate the total dilution. The first dilution occurred when 25 grams of sample was added to 475 milliliters of 0.1% peptone and homogenized. Then another dilution occurred when 10 milliliters of the homogenized solution was transferred into 90 milliliters of 0.1% peptone solution. The next dilution occurred when 0.1 milliliters of solution was transferred to 9.9 milliliters of 0.1% peptone, which is a 100 times dilution. Finally, 0.1 milliliters of the diluted solution was spread plated. This represents another 10 times dilution. Now that we have all our individual dilutions determined, we can simply multiply all of them together to obtain our total dilution. Then use 1 divided by the total dilution to calculate the total dilution factor. To calculate the CFU per gram, first average the duplicate colony counts for each technical replicate. Then use the average colony count divided by the total dilution, or multiply the colony counts by the total dilution factor. Follow the same steps to calculate the CFU per gram for all other biological replicates, and use these values for statistical analysis. Thank you for watching.